Yeah, so there were two presentations at ASH that give a hint about how we may augment the effect of glofitamab. Roche has two co-stimulatory bispecific antibodies in development. Now, we know that CAR T cells really broke through, not just through binding of the T cell, through activation of the T cell receptor, the CAR T receptor, but also through incorporation of a co-stimulatory domain within the CAR T receptor that really led to the products that we're used to using today and which have been registered for treatment of large B-cell lymphoma. And at the moment, glofitamab works essentially through the CD3 T-cell receptor. The question that we've been asked and which we've presented in trials in progress presentations at ASH is whether a second co-stimulatory signal might improve the activity of glofitamab. Now, mature data wasn't presented at ASH. These were trials in progress, but nevertheless, I think interesting in terms of strategy. There are two molecules that were presented. One is a CD19 4-1BB ligand bispecific antibody. And this is given either with obinutuzumab as a monotherapy or given with glofitamab with the typical phase 2 go forward dose. And so essentially the monotherapy treatment has been established as being safe, and we presented that data at ASH. And now the combination therapy is still being evaluated. And so the question that trial is asking is if we add a second signal, 4-1BB and the new bispecific antibody in combination with glofitamab, and through providing that second signal augments T-cell activity, whether we see more anti-cancer efficacy compared to glofitamab alone, and whether that can be delivered safely is the question that clinical trials are asking. It's sort of tantalizing because 4-1BB is also present not just on T cells, but also on other cell populations like NK cells. So that's that co-stimulatory domain that's being tested with that novel antibody. The other novel antibody that's being used in combination with glofitamab is CD28, CD19 bispecific antibody. The principle is very similar, providing a second signal. This trial is interesting because it evaluates both intravenous and subcutaneous delivery of the additional agent. Both trials are ongoing. Patients have been treated. So far, the safety signals are good, but we don't yet have advanced data to present on efficacy. But we know from what's been presented at ASH that data is coming very soon. And these trials are actively recruiting at sites globally.